Good morning, students. Today, in a new chapter of Principle of Inheritance and Variation, we are going to study Mendel's work and monohybrid cross. Before proceeding towards the chapter, let us understand few definitions. First of all, inheritance. It is the process by which characters are passed from parents to the offspring. As we know, we inherit certain traits from our mother and certain traits from the father. So, this process is called as inheritance. Variation is the degree by which progeny differ from parents. Like, we also differ from our parents, we also differ from our mother and from our father. And the reason behind this variation is the sexual reproduction. We all know that in asexual reproduction, organisms arise from single parent and no variation is seen in those organisms. Genetics is a branch of biology which deals with inheritance and variation of characters from parents to offspring. Now let us move ahead. Gregor Johann Mendel. He is popularly known as father of modern genetics. He was born in Austria and conducted his experiments between year 1856 to 1863. His experimental organism was Pisum sativum, the garden pea plant. He worked in a monastery as an abbot and worked in the garden of that monastery and proposed certain laws of inheritance and variation. Now we will see how Mendel proceeded with his work. Mendel selected garden pea plant and in that pea plant he studied 7 contrasting traits, and these traits were flower color, plant height, seed color, seed shape, pod color, pod shape and flower position. In these traits he observed that certain traits are dominant, which are found in larger number of population, while certain traits are recessive, which occur in lesser number of population. Mendel started his work by taking true breeding lines. These are those plants who have been generated through repeated self-pollination and have been homozygous for a particular trait. That means no variation was seen. So you can see here a tall pea plant when self-pollinated always produces tall pea plant. Now coming to his work. Mendel took the plant in which it was producing green pod and yellow pod. So we have understood the concept of true breeding lines. Here Mendel selected true breeding lines. On the crossing of these two plants, Mendel observed that progeny was showing only green color pods. Then Mendel self-pollinated in these plants and there he observed certain variation. He found that 3/4 of progeny was showing the green pod while 1/4 of the progeny was showing yellow pod. So this led Mendel think what is the reason behind it. And Mendel also performed experiments on the other traits also to look whether this is the same situation with other traits or not. So here you can see Mendel performed the experiment on flower color, flower position, seed color, seed shape, pod color and stem length. In every character, he found the same kind of situation, that approximately three-fourths of the progeny was showing one trait and one-fourth was showing another trait. One more thing to observe in this diagram is you can see the number of sample which he studied. See this huge number. This because we all know that from studying a large sample size, our data becomes more accurate. So Mendel performed the experiment while taking large sample size. So what he concluded from his work? He said something passed unchanged from parents to offspring through gametes, and these are called as factors. These factors are distinct and do not blend. As we saw earlier, when Mendel crossed green pod with yellow pod, there was no intermediate color, while in the progeny, there was only one color, and that was green. Nowadays, we call these factors as genes. Genes are unit of inheritance, and these bring characters from our parents into us. Genes which code for a pair of contrasting characters. These are called as alleles. Mendel also proposed certain terminology. He gave certain alphabetical symbols for the alleles. And in a diploid organisms, two alleles are present. Dominant allele is always written in capital letter, and it is the allele which expresses itself in a pair of dissimilar traits. Recessive alleles do not express itself in a pair of dissimilar trait, and it is written in small letters. In homozygous individuals, alleles are similar, while in heterozygous situation, alleles are different. Genotype is a genetic makeup of organism. That means what kind of alleles are present is shown in genotype, while phenotype is observable trait. As we see that pod was green color or in yellow color. Another rules were, here you can see earlier we studied that dominant trait is shown with the capital letter while recessive trait is shown in the small letter, right? So if seed shape is round, then in wrinkle we will write it as a small letter. Seed color, the dominant color is yellow. The pea seeds which we generally consume in winter, that is green in color because it is unripe seed. When it will be let the ripen on the plant, then its color will change into yellow. So these are the various symbols of the characters which Mendel studied in his experiment. Now coming to his work again, when Mendel crossed tall plant with a short plant, then all offspring were of tall. There was no blending of trait, no intermediate height was observed. Tallness, the allele responsible for this, was completely dominant over the recessive one. But when Mendel took these plants and did the self-pollination, then he observed that three-fourths of the progeny was showing dominant trait and one-fourth of progeny was showing recessive trait. Now Mendel explained this thing with the help of alleles. Now let us understand that. Mendel took one trait at a time. that is plant height. Alleles responsible for the tallness was capital T, and for the dwarfness it was small t. When he crossed the tall plant and the dwarf plant, he observed the progeny, which was named as F1. That means first filial generation. So result, let us see. First of all, gamete formation. These are the alleles which got segregated during gamete formation. Here is the fusion of gametes and result. So result was that offspring resembled only one of the parent and there was no intermediate trait. Genotype of every plant was capital T and small t, and phenotypically all plants were tall. Now, to study the F2 generation, that is second filial generation, Mendel did self-pollination in the plants of F1. Now let us understand this. Gametes. This is the gamete formation for both of the parents. Fusion of gamete, and here we can see that out of the four, three individuals are tall and one is dwarf. But one thing to note here is that this is homozygous, while this and this, these both are heterozygous. Result. Offspring were either tall or dwarf, but not intermediate. Genotypic ratio that we have seen here, it is 1:2:1. That means 25% of individuals were homozygous tall, 50% of individuals were heterozygous tall, while 25% of individuals were dwarf. And phenotypic ratio of tall is to dwarf is 3:1. That is 75% of individuals were tall and 25% of individuals were dwarf. Now let us study one more monohybrid cross. This time we will study seed color. We know that dominant trait is yellow and recessive trait is green. And dominant trait is shown by capital letter and recessive trait is shown by small letter. Right? So these are the alleles responsible. Parents. So we can make a Punnett square very easily. For the first filial generation, gamete formation, fusion of gametes, and result. So in this we find that no intermediate trait is observed and all plants are producing yellow color seeds. Now performing F2. Now coming to make cross for F2. Trait is seed color. We know the alleles responsible behind it. Parents for the F2 are individuals of F1. Now let us make Punnett square. First of all, gamete formation, fusion of gametes, and now look at this result. 3/4 of individuals are showing yellow color seed, while 1/4 of the plant are showing green color seed. Out of this yellow color seed, 25% are homozygous yellow, while 50% are heterozygous yellow, and only 25% are showing green color, and these are homozygous. With this work, Mendel concluded certain laws, and these are Law of dominance. In a heterozygote, one allele dominates the another and masks the expression of recessive allele. As we studied, capital T was masking the trait of small t, that's why plants were tall, and capital Y was masking the trait of small y, that's why seeds were yellow. Another important law was law of segregation. During gamete formation, allele for each gene segregate from each other so that gamete carries only one allele. As we know, we are diploid organisms and gametes produced by people are always haploid. That means they carry only single set of chromosomes and they have only one allele of the one homologue. So here we can see that if this is the individual's genotype, then while gamete formation, one gamete will be having capital T and another gamete will be having small t. Now coming to assignment. When a tall pea plant is self-pollinating, 25% of progeny is dwarf. What is the genotype of plant? I hope you can draw the Punnett square and you will be able to solve it. What do you understand by true breeding lines and what are the different laws derived from this cross? All are clear in the content studied. So wish you all the best. Thank you.